animal cell and plant cell. And if you remember, I talk about the human cell is a type of animal cell, okay? And I talk about the cell component like the cytoplasm and the plasma lemma and the nucleus, okay? That forms the eukaryotic cell. And I talk about the plasma lemma cell membrane, and also I talk about some organelle like mitochondria, ribosome, and Golgi and the endoplasmic reticulum, okay? And now I talk about the Golgi apparatus and the other organelle in the cytoplasm of the human cell, okay? What is the Golgi apparatus? Look at this picture. The Golgi apparatus is formed by some parallel sac or cistern within the cytoplasm. Usually the Golgi apparatus is located in one side of the nucleus, okay? And it's involved to the protein synthesis, okay? The dynamic organelle called Golgi apparatus or Golgi complex complete post-translational modification of protein synthesis, okay? So it's involved to the protein synthesis and the post-translational modification. That means when the protein or polypeptides chains is formed within the endoplasmic reticulum with help of the ribosome, for example, this polypeptide enters to the Golgi apparatus and alters and modifies to the final shape of the protein within the Golgi apparatus, okay? This protein synthesized in rough endoplasmic reticulum and then packages and addresses these proteins to proper destination, okay? The final destination of this protein is about, for example, the proteins for the plasma lemma, the protein or enzyme for the lysosome, or the protein for the secretion of the cell, okay? And name of the Golgi apparatus is about the histologist Camillo Golgi. He discovers the Golgi apparatus in 1898, okay? The Golgi apparatus is composed of smooth membranous saccles containing enzymes for this function. Look at this picture. Usually the Golgi apparatus has a concave side. It's called cis, and this concave side usually is located near the nucleus of the cell, and in the other side it has a trans face. It's convex, okay? Near the saccule of the Golgi apparatus you can see some vesicle that transport the content or proteins from one saccule to the other saccule of the Golgi apparatus, okay? So it's important. The Golgi apparatus is important for post-translational modification of the proteins, is important for protein synthesis, okay? Look at this picture again. This picture shows the schematic shape of the Golgi apparatus, and this is a photography, electron photography of the Golgi apparatus under the electron microscope. You can see the parallel cistern and some vesicle that located near these saccles, okay? Totally, the function of the Golgi apparatus is modification, packaging, and sorting, okay? Modification of the proteins that enters to the Golgi apparatus from the rare rough endoplasmic reticulum. Okay, the Golgi apparatus can modify new proteins destined for lysosome, for secretion, and plasma membrane. Okay, and the second function of Golgi apparatus is packaging. Packages enzyme for lysosome, for example, and protein for secretion. And sorting, it can sort all materials for lysosome, secretion, and incorporation into the plasma membrane. Usually three different destination is about the protein that's sorted by the Golgi apparatus. Okay, the enzyme for lysosome, the proteins for secretion like hormones, or the protein for the plasma membrane. Okay, and this picture is electron photograph of the parallel suckle of the Golgi apparatus. Under the electron microscope, you can see these suckles, okay, or sucks. And this is the schematic photograph of Golgi apparatus. Look at here. The proteins forms the protein for secretion, for example, synthesized within the rare, rough endoplasmic reticulum, with help by the ribosome, if you remember. And from the rare, these primary proteins enters to the Golgi cisterns, okay? And you can see here this vesicle is called transporting vesicle, can transport the protein from the first suckle to the other suckles, for example, okay? And some proteins can help to this transport. For example, the protein COP2 can help to the transport of the vesicle in anterograde, for example, from the nucleus to the apical of the cistern of the Golgi apparatus, and the protein COP1 can return this vesicle to the first suckle of the Golgi apparatus, for example. Okay, and finally you can see here the three different destinations are for the proteins: lysosome, enzyme for lysosome, the protein for secretory granule for secretion or exocytosis, and the proteins for the plasma membrane, like the integral protein, like the receptor, for example. It's located finally in the plasma membrane. Okay, this is the function of the Golgi apparatus, and it's very important for protein synthesis. Okay, the other organelle it's live within the cytoplasm is lysosome. Okay, lysosome is a vesicle. Okay, it has a membrane. It's enclosed by the membrane, and it contains the enzyme for digestion within the cytoplasm. So the lysosome is important. The enzymes for the lysosome is produced by the rare rough endoplasmic reticulum and packaged and sorts within the Golgi apparatus, and finally enters to the lysosome. Okay, the lysosome are sites of intracellular digestion and turnover of cellular components. Okay, that means the firing part, the old organelle of the cytoplasm, can hydrolyze within the lysosome. Okay, it's called intracellular or turnover of intracellular component. The lysosome are membrane-limited vesicle that contains about 40 different hydrolytic enzymes. 40 different hydrolytic enzymes, okay? Many of the enzymes are located within the lysosome, like lipase, protease, amylase, and the other enzymes are located within the lysosome, okay? And the enzyme within the lysosome becomes activated in low pH, okay? So the membrane of the lysosome contains some pumps for the active pumps for transfer the protons within the lysosome, and when the protons H+, for example, it enters to the lysosome, the pH within the lysosome decreases, and the enzymes become active, okay? Also, the nature and activity of the lysosomal enzyme vary dependent on the cell type. The most common are acid hydrolysis, such as protease, nuclease, phosphatase, phospholipase, sulfatase, and the other enzyme, okay? As can be seen from this list, lysosomal enzymes are capable of breaking down most macromolecules, okay? So the different macromolecules that enters to the cells or the old organelle can hydrolyzes within the lysosome by help the lysosomic enzyme, okay? This picture shows the macrophage cell. The macrophage cell usually is located in connective tissue, and it can hydrolyzes and can phagocytize the foreign particle that enters to connective tissue, for example, and the cytoplasm of the macrophage cell contains many of lysosome organelle with the high activity of the enzyme, okay? You can see the many abundant of the lysosome in cytoplasm of the macrophage cell. And this picture shows some of this lysosome within the cytoplasm of the macrophage, and you can see the mitochondria near this lysosome within the cytoplasm of the macrophage cell, okay? The enzyme of the lysosome are very important, and the lack of this enzyme usually leads to the disease, a specific disease. For example, the Horler syndrome. The Horler syndrome is about the skeleton and nervous defect, okay? It's about the lack of the alpha L iduronase. That means this enzyme is destroyed, and this syndrome is occur within the body, okay? So the enzyme of the lysosome is very important. This picture shows the first lysosome or primary lysosome that's formed by the Golgi apparatus, by the rare rough endoplasmic reticulum, and Golgi apparatus, for example, forms the enzyme. The enzyme is proteins, okay? You know that the enzymes are proteins, and the proteins produced by help the rare and the Golgi apparatus, and package and sorting for the lysosome, for example, within the cytoplasm, okay? This called the primary lysosome. When the foreign particle like the bacteria phagocytoses and enters to the cytoplasm of the cell, this vesicle is called heterophagosome. And when the heterophagosome fuses with the membrane of the primary lysosome, this lysosome is changed to the secondary lysosome. The enzyme of the secondary lysosome becomes active and hydrolyzes the bacteria, for example, and the macromolecule of this organism hydrolyzes to the subunits, for example, to amino acids and glucose and the other lipids, fatty acids, for example. These molecules enter to the cytoplasm, and the cytoplasm of this cell can use of these subunits of macromolecules. But some part of the foreign particle, like the bacteria, okay, cannot digested by the lysosome, and usually this part of these particles, for example, remain within the cytoplasm as the residual body, okay. Lipids. Hello, dear student, can you hear me? Okay, for a moment the connection is cut, okay? I share again. Indigestible material is retained within a small vesicular remnant called the residual body. That means this material, okay? In some cells, in some long-lived cells, for example, the neurons, the heart muscle cell, okay, that had a long lived okay, and these cells are the permanent cells and don't have the division, for example. Within the cytoplasm of this cell, the residual body can accumulate, the residual body, this body. The residual body is indigestible particles or molecules, for example, okay? Become accumulate and forms a pigment, for example, it's called lipofuscin granule. Okay, so we can see the lipofuscin pigment granule within the permanent cell like the neuron cell and the heart muscle cell, okay? This picture shows the lysosome, and within the lysosome we can see a mitochondria, and when the lysosome hydrolyzes the old organelle within the cytoplasm, this vesicle is called autophagosome, okay? And when the lysosome hydrolyzes the foreign particle is called heterophagosome. Okay, so this is the difference between the autophagy or autophagosome and heterophagosome or heterophagy. Okay, the other particle or the other organelle within the cytoplasm is called protozoan. Protozoan. Look at the protozoan. This is the shape of the protozoan. It's cylindrical. It's like a cylinder, okay, or cylindrical shape. The protozoan don't have the membrane, okay, and it's very small. It's very small particle or organelle within the cytoplasm, and it's involved to hydrolysis of the proteins. Which protein? The denaturated proteins or destroyed proteins. Okay. Sometimes the free polyribosomes, okay, and ER bound polyribosome. If you remember, there are two types of the polyribosome within the cytoplasm. Free polyribosomes involves to production of proteins with destiny for the proteins for the cytosol or the cytoskeleton, for example. Okay, and the ER bound polyribosomes are involved to production of the protein for secretion for the exocytosis, for the lysosome, for example. Okay, for the secretory vesicle or the lysosome, for example. Okay. And during the synthesis of the proteins, sometimes some proteins become destroyed and becomes misfolded during the folding and the translation or post-translational modification. Okay, become destroyed or denaturated proteins. A polypeptide within the cytoplasm, it's called the ubiquitin. The ubiquitin can distinguish the misfolded or denaturated proteins, and the ubiquitin leads these proteins to the protose. Okay, the protose is cylindrical, and in one of the protosome, attention please, in one of the protosome, there are some protease enzyme can cut the protein chain, okay, and finally it can hydrolyze the polypeptide to the peptide or amino acid. This amino acid that release in the other side of the protosome can use for production of the new proteins. Okay, this is the function of the protosome. The function of the protosome. Protosomes are very small, abundant protein. Okay, the structure of protosome is protein. Okay, is protein complex not associated with membrane. That means it's don't have the membrane. Okay, each approximately the size of the small ribosomal subunits. Okay, and they function to non-functional polypeptide. Okay, so it can hydrolyze the misfolded or denaturated protein within the cytoplasm. It's very important for the cytoplasm. Okay, organelle within the cytoplasm is called peroxisome. Peroxisomes are very small, spherical, membrane limited organelle, okay, or membranous organelle. In the past, we called the peroxisome as microbody because of this is very small. Okay, so the peroxisome or microbody containing the enzymes that use the oxygen, like the mitochondria. If you remember, within the cytoplasm, the other organelle uses the oxygen is mitochondria. But when the mitochondria uses the oxygen, it can produce the energy. Okay, it can produce the energy like the ATP molecule. But when the peroxisome uses the oxygen, okay, it cannot produce the energy. And usually the peroxisome can oxide some material and can modify the structure of some toxins, for example, and some drugs, for example. Okay, so it's important for the cytoplasm of the specific cell, for example. Okay, so that use the oxygen to remove the hydrogen atom. Okay, but uses the peroxisome, but uses the oxygen, it can remove the hydrogen atoms or hydrogen molecules from fatty acids in a reaction that produces hydrogen peroxide. Okay, H2O2. H2O2 is toxin for the cells. It is toxin, okay, and it's very dangerous, and it must broken very quickly. Okay, and the H2O2 must be broken down to water and oxygen again by the catalase enzyme. Okay, this is the function of peroxisome. The peroxisome is important. Some cells, for example, the hepatocyte cell can hydrolyze the alcohol, the toxin, it can detoxification, these cells can detoxification some toxin, some drugs, some alcohol. Okay, and in this picture you can see some secretory granule that accumulate in the apex of the secretory cells. Okay, the other part of the cytoplasm is cytoskeleton. Attention please, I must open the other file. Okay, the other structure in the cytoplasm is cytoskeleton. Look at this picture. This picture shows the cytoskeleton, and we use the antibody against the proteins of the cytoskeleton. So you can see only the proteins that form the cytoskeleton. Okay, and we cannot see anything within the cytoplasm, or we cannot see the nucleus, the cell membrane. See the structure of cytoskeleton. The cytoskeleton is important for the cell because these proteins can determine the shape of the cell. Okay, in this cell you can see this framework, these proteins forms the shape of the cell. So this structure is called cytoskeleton. Okay, the cytoplasmic cytoskeleton is a complex array of three structure: microtubule, this picture, microfilament, and intermediate filament. Microtubule, microfilament like the actin filament. You hear about the actin filament previously, okay? And intermediate filament, okay? These proteins forms the shape of the cells, okay? Form the shape of the cells, or form some accessory of the cells like the cilia or flagella, for example, and helps the movement of the cells, okay? And the cytoskeleton involves the movement of the cells, movement of the molecule within the cytoplasm, or movement entirely of the cell, okay? So the protein polymers determines the cytoskeleton proteins, okay, determine the shapes of cells, and the cytoskeleton play an important role in the movement of organelles and cytoplasmic vesicle, okay. Also allows the movement of entire cells. So the cytoskeleton is important within the cytoplasm of the human cell, okay. This picture shows the different type of the cytoskeletal structure, like the microtubule, that means it's like a tube, okay, in a very small size. It's called microtubule. Microfilament, like the actin filament and intermediate filament. The general function of the cytoskeleton. It has two roles or two functions: structural and movement. Structural, that means the cytoskeletal proteins provide the structural support to cells, okay, and stabilize junction. The cytoskeletal proteins involve some junction between two cells. And the second general function is movement, assist with cytosol and cell motility or cell movement, okay. The first structure is microtubule. Look at here. This is the microtubule. Each microtubule is hollow tube or microtube, okay? It's hollow with an outer diameter of 25 nanometer. That means this diameter is a nanometer. Each microtubule, it has two ends, plus end and minus end, okay? Plus end and minus end. And it's formed usually by 13 protofilament. 13 protofilament. Each protofilament is formed by a heterodimer alpha-beta tubulin subunits. This subunit alpha-beta tubulin polymerized in plus end, okay, and the microtubule can elongate it, okay, can bruise and can elongate it from the plus end by polyreflection of the alpha-beta tubulin, okay. Microtubules vary in length but can become many micrometer long, okay. Microtubules are organized into a larger array called axoneme. Axoneme is a pattern into the cytoplasmic extension. For example, within the cilia and flagella, we can see the axoneme pattern of the microtubule that forms the structure of cilia and flagella, for example, okay? And Usually the minus end of the microtubule is attached to the other structure. It's called the microtubule organizing center, like the centriole, for example. Okay, look at here. The subunit alpha beta tubulin can added in the plus end and can proliferate it and elongate the microtubule. Okay, the protein subunit of a microtubule is a heterodimer. Alpha beta tubulin, alpha and beta tubulin. Okay, and a total of 13 units are present in one complete turn of the spiral. It's added spirally, and finally it forms the microtubule. Okay, in this picture you can see the axoneme pattern. Okay, in axoneme pattern we can see a formula of 9 plus 2 of microtubule. The 9 doublet microtubule is located in axoneme, and we can see this pattern within the cilia or flagella. Okay, and that microtubule is formed by microtubule A and microtubule B that share three of this filament. Okay, it share 3 of these filaments for junction of these doublet microtubule. And usually the microtubule A has 2 arms. This arms is formed by protein dynein. The protein dynein is the ATPase, can hydrolyze the ATP, and it uses the energy for sliding the neighbor microtubule, and this action it's help the movement entire of the cilia and flagella. Okay, so the cilia and flagella have the movement, okay, and can help the movement of the cell, for example. Within the cytoplasm, the cilia or flagella, for example, is attached to the other structure. It's called basal body. The basal body is formed by 9 triple or triplet microtubule. You can see here the triplet microtubule. Okay, this structure is called microtubule organizing center or MTOC because polymerization of the tubulins is directed by microtubule organizing center. So you can see the different arrangement of the microtubule in a structure, in different structure in the cytoplasm of the cell. Okay, all of this structure formed by the microtubule. This picture shows a cilia. Okay, a cilia. Attention please, a cilia is covered by the plasma membrane. It's covered by plasma membrane, and if we prepare a section of the cilia, we can see the axoneme pattern. Axoneme pattern. The formula of 9 doublets plus 2 central microtubule. 9 plus 2. Okay, this is the formula of axoneme. And look at here. In the cytoplasm, the cilia attach to the basal body. And look at here. The basal body is formed by 9 triplets of microtubule. that connect to each other by some proteins, for example. This pattern of basal body is similar, is like to the centriole. The centriole is directed the formation of the mitotic spindle. Mitotic spindle is formed by microtubule during the cell division. When the mitotic spindle is formed, the mitotic spindle is formed by microtubule, okay? The other function of the microtubule, within the cytoplasm, this microtubule helps to the movement of the organelles, okay, within the cytoplasm, okay? Transport of the organelle or vesicle along microtubules is under the control of some proteins called motor proteins. Okay, so some proteins is called motor proteins. Motor proteins helps the movement of the vesicle or organelle within the cytoplasm. Okay, this motor protein uses the energy, uses the ATP, okay, in moving the larger structure. One of the motor proteins is kinesin. Kinesin, this protein, for example, this is the schematic shape of the kinesin protein, and this is the microtubule. Look at here, this end of microtubule is the minus end, and this end is plus end, okay? The kinesins carries the material away from the MTOC, that means from the minus end near the nucleus toward the plus end of microtubule, okay? This movement is called anterograde transport from the minus end. The minus end usually is attached to the MTOC organelles, okay? And the opposite direction, okay? The opposite direction. Attention please, the cytoplasmic dynein, the dynein protein. Attention please. In axoneme, if you remember, the dynein arm attached to the microtubule A, but the cytoplasmic dynein, the other type of the dynein, carries material along microtubule in the opposite direction. That means from the plus end to the minus end. Okay, it's called retrograde transport. So, some proteins that call the motor proteins can transport the vesicle or organelle within the cytoplasm. Okay, from the around the nucleus to the, for example, to the periphery of the cytoplasm by the kinesin, and the opposite direction is formed by the dynein. This picture shows the centriole. Attention please, the other MTOC organ is centriole. Probably you hear about the name of the centriole. When the cell ready to the division, for example, the centriole organelle, the two cylindrical and a small, very small organelle appears near the nucleus of the cells, okay? And this double cylinder is separated to each other and move to different pole of the cytoplasm, and between this centriole, the mitotic spindle is formed and it's closed by the microtubule. Okay, so the centriole that forms by the microtubule, the pattern of the structure of the centriole is the 9 triplet of the microtubule. Okay, it's very important for cell division. It's very important for cell division. Okay, the dominant MTOC is most somatic cell is the centrosome, which is organized around two cylindrical centriole. The centriole and the cytoplasm around the centriole is called centrosome. Okay, each about 0.2 micrometer in diameter and 0.3 to 0.5 micrometer in length. Each centriole is composed of 9 highly organized microtubule triplets. This is the structure of the centriole, and you can see the other picture of the centriole during mitosis division. During mitosis division, the centrosome divided into halves, okay, which move to opposite pole of the cell. I talk about this, okay, and become organizing centers for microtubule of the mitotic spindle. Okay, this picture shows the cytoplasm of the cells. Within the cytoplasm we can see the microtubule. You can see the microtubule that form the shape of this cell. Okay, and this picture shows the cilia at the apex of the respiratory epithelium. All of these are cilia, and the structure of cilia is the axoneme. Okay, it's formed by the microtubule. Okay, the other component in cytoskeleton is microfilament. The microfilament composed of actin protein. The actin protein allows cellular motility and most contractile activity in the cell, okay, by reversible assembly of the actin filament and interaction between this filament and the associated protein myosin. Usually you hear about the actin filament and the myosin filament within the muscle cell. Okay, within the muscle cell, the interaction between the actin and myosin is formed some structure for contraction. Okay, but the actin filament that forms by the F-actin 2 or double F-actin strand, the subunit of these is called G-actin. Okay, by proliferation of the G-actin, the F-actin is formed, and 2 F-actin usually forms the actin filament. Like the microtubule, the microfilament actin has the minus end and the plus end, and proliferation of the subunits is formed in plus end. Okay, the actin filament usually is located under the plasma lemma. Attention please, it's located usually under the plasma lemma, and by some proteins is attached to the integral proteins. Okay, and it helps the shape of the cells. It's important for the stability of the cell membrane, for example. Okay, and it's helped the cytoplasmic division during the cell division, and within the cytoplasm we can see the actin filament is distributed within the cytoplasm. Okay, the filament of actin, they are composed of globular G-actin. the G-actin, the globular G-actin monomer that assemble in the presence of potassium and magnesium into a double strand helix of filament. It's called F-actin. Each of filament is called F-actin, and F-actin is formed by proliferation of the G-actin, okay, the subunit. And in this picture you can see the cell membrane. This is the cell membrane, and these are the integral proteins. And here is the cytoplasm, and you can see the actin filament attached to the integral protein by some proteins like alpha-actinin, vinculin, taurine, and the other proteins. And the actin filaments support the cell membrane. Okay, it's very important for establishment of the cell membrane. Actin is very abundant in all cells, usually concentrated as network of actin filaments and abundant free globular G-actin subunits concentrated near the cell membrane. Okay, a region sometimes called of the cell cortex, and in cellular extensions, okay, like the microvillus, for example. And here you can see some strand of the actin filament. It's attached to the plasma lemma, or it can organize in parallel strands, for example within the microvillus, and usually within the cytoplasm the actin filament can crosses to each other, and some protein links these filaments and forms the complex structure of the actin. The actin filament, like the macrotubule, can help the movement of the organelle within the cytoplasm. Okay, in this picture you can compare the thickness of the microtubule. Look at here, the microtubule is like a small tubule, and this is the filament. It's like a line. Okay, you can compare the thickness of the microtubule and microfilament under the electron microscope. Okay, and finally the intermediate filaments. Intermediate filaments are the other type of the filament that forms the cytoskeleton. Are strong proteins, strong filaments. Usually the intermediate filament can help the cell adhesion and within the special cell like the epithelial cell. Some of these filament like keratin, for example, you can maybe hear about the keratin protein. Keratin protein fills the cytoplasm of the epithelial cell in epidermis of the skin, for example, or it can find within the hair or nail. Okay, is a strong protein. Or cytokeratin are a diverse family of acidic and basic isoform that compose a heterodimer subunit of intermediate filament in all epithelial cell. Okay, the intermediate filament are much more stable than the microtubule and actin filament. Moreover, these filaments are composed of different protein subunits in different cell type. Okay, this table shows the different intermediate filament within the different type of the cell. For example, you can see the cytokeratin or keratin within the epithelial cell. You can find the other intermediate filament. It's called desmin, In muscle cell, we can find desmin. Or in astrocyte cell, the astrocyte cell are a type of the neuroglial cell. Within the astrocyte, we can find the GFAP protein, and in the neuron, we can find the neurofilament, for example, and the other intermediate filament are located in different type of the cell. At the end of chapter 2, of John Coelho histology, we reach to some inclusion vesicle, okay, or particles. The cytoplasmic inclusion have little or no metabolic activity, which distinguish them from organelle. For example, there's secretory granule. The secretory granule can accumulate in apex of the secretory cell, or the accumulation of the glycogen within the cytoplasm, or the other material like the lipids. For example, you can see the lipid droplet within the cytoplasm. All of these structure, all of these component, totally it's called inclusion material, okay? You can see here the aggregation of the glycogen molecules within the hepatic cell. And in this picture, you can see the lipofuscin molecule, the pigment material. Okay, the chapter 2 is finished. You can rest for 5 minutes. Okay, after 5 minutes, I will continue, and we must learn about the nucleus of the cell. Okay, and be interested to the chapter 3. Rest for 5 minutes. Okay. Dear student, good morning again. Are you ready? Okay, in this lecture, we learn about the nucleus of the cells, okay? Chapter 3 of basic histology. Tranquilla basic histology, okay? So this lecture is about the chapter 3, the nucleus, okay? Okay, look at here. What is the nucleus? Nucleus is a large organelle within the cytoplasm, okay? The nucleus frequently appears as a large, rounded, or sometimes oval structure, often near the center of the cell. Attention please, the shape of the nucleus is dependent the shape of the cell. For example, in flat cell, the nucleus becomes flat. In cuboidal cell, the shape of the nucleus is rounded, is spherical, okay? And in lung cell or the columnar cell, for example, the shape of the nucleus becomes oval, okay? And usually the nucleus is located at the center of the cell, but sometimes it's located near the base of the cytoplasm, for example, okay? And some cells has more than one nucleus, okay? But usually each cell has one nucleus. The nucleus also contains the molecular machinery to replicate the DNA. That means the nucleus is houses of genes, the house of genes, the DNA, okay? And it has the molecular machinery for synthesizes and process all type of the RNA. For example, the messenger RNA is replicated from the DNA, okay? What is the function of the nucleus? Attention please. The cellular regulation. That means the nucleus of the cell can regulate all activity of the cell. Okay, it can regulate all activity of the cell. It hosts genetic material which directs all cellular activity and regulates cellular structure. The second function is production. What does this mean? The nucleus can produce subunit of ribosome, if you remember. Produce ribosomal subunits in nucleolus. Attention please, the nucleus contains the nucleolus. This very small and dense structure is called nucleolus. Nucleus and nucleolus, okay? And the nucleolus produce the ribosomal RNA, okay? So the second function of the nucleus is the production of ribosomal subunit. Contains a three-part Nuclear envelope, the membrane that surrounded the nucleus, is called nuclear envelope. 2. A mass of DNA. A mass of DNA and associated proteins. DNA and associated proteins called chromatin. That means the nucleus filled by chromatin. Okay? And a specialized subdomain is called nucleolus. Nucleolus, okay? Nuclear envelope, chromatin, and nucleolus. This picture shows the hepatocyte, and you can see the nucleus of the nucleolus within the nucleus of this cell. Okay, and this picture shows the schematic shape of the nuclear envelope. Attention please. Attention please. The nuclear envelope contains two separated membrane, outer membrane and inner membrane, and the prenuclear cistern. Between two membrane, this space is called prenuclear cistern or prenuclear space. The outer membrane of the nuclear envelope continues with the rough endoplasmic reticulum. So you can see the ribosomes are attached to the outer surface of the nuclear envelope. Okay, the nucleus forms a selective permeable barrier, can regulate the transport of the material. Okay, that means the nuclear envelope don't allow the enter each of the material to the nucleus. Okay, it select the material, and the material usually pass through the nuclear pore. Attention please, where the outer and inner membrane of nuclear envelope reach to each other forms the pore. This pore is called nuclear pore. Okay, and look at here. The inner surface is nuclear lamina. The lamine protein or nuclear lamina is the other type of the intermediate filament of the cytoskeleton, if you remember. Okay, lamina, closely associated with the inner nuclear membrane, is highly organized meshwork of protein called nuclear lamina. Lamine or lamina, okay, it supports the inner membrane of the nuclear envelope. And in this picture you can see the outer membrane of nuclear envelope continues with the rough endoplasmic reticulum. And here you can see the nuclear pore. The nuclear pore usually fills by some proteins called nuclear pore complex. And in this picture you can see this complex. That means some proteins fills this pore, and these proteins allows only the specific molecule to enters to the nucleus. Okay, the inner and outer nuclear membranes are breached at nuclear pore, here at nuclear pore complex, and various core proteins of nuclear pore complex called nucleoporine. These proteins called nucleoporine, and nucleoporines arranged like the basketball basket, for example. You can see this shape. proteins can help the transport of the molecule. For example, importing protein helps to entrance of the molecule within the nucleus, and exporting molecule can help the exit of the molecules from the nucleus to the cytoplasm. And here you can see again the nuclear lamina. Nuclear lamina can attach to the inner surface of the nuclear envelope. The nuclear lamina is a site for attachment of the chromatin. Look at here. Is a site for attachment of chromatin within the nucleus. Okay. This picture shows the electron photograph of nuclear pore. Look at here. You can see the outer and inner layer of the nuclear pore, and here is nuclear porine. And this picture shows the different techniques for showing the nuclear pore. It's called freeze fracture. At first, the nucleus or the cells is freezed, and then it's breakdown or fractures, and we can see this shape. Okay, and you can see the double membrane and nuclear pore. And in this picture you can see again the nuclear porine complex at the surface of the nuclear pore. The other component of the nucleus is chromatin. What is the difference between chromatin and chromosome? Do you know? What is the difference between chromatin and chromosome? Attention please. The chromatin is formed by DNA and the specific proteins. Okay? But in non-dividing cells, we can see the chromatin within the nucleus. But when the somatic cells, for example, becomes ready to division, the chromatin compacts and forms the chromosome. Okay, so the chromosome is a compact DNA or compact chromatin. Look at here, within the nucleus of the cells, we can see usually two different types of the chromatin. Euchromatin is a light chromatin, is an open chromatin, and heterochromatin. Heterochromatin is a closed or packed chromatin. Okay. In non-dividing nucleus, chromatin consists of DNA and its attendant proteins in a largely uncoiled state. Two types of chromatin can be distinguished with both the light and electron microscopy, heterochromatin and euchromatin. Okay. This picture shows a chromosome. Each chromosome has two arms. Okay. It's formed by two chromatid. Okay. Chromatid. Each chromatid is attached in centromere here to each other. The chromatid attached to each other in centromere here. Okay. And if we open the chromosome like this, we can see the subunits that forms the chromatin or chromosome. And you can see here the DNA strand is turned around the histone proteins, octamer histone proteins that form the nucleosome. Nucleosome are subunits of the chromatin and chromosome. Okay. DNA in chromatin is extensively packaged by association with basic protein called histone. Okay, these proteins are histone proteins, and with various non-histone proteins. The structural unit of DNA and histone is the nucleosome. So each of these are nucleosome. The subunit or structural unit of the chromosome and chromatin is nucleosome. The nucleosome has a core of 8 small histone. 8 small histone that DNA strand turns around the histonic proteins and form the nucleosome. Look at here, you can see the 8 proteins is formed by double H2A, H2B, H3 and H4. It's formed the histone core of the nucleosome, and you can see the DNA strand turns around this octamer structure and reach to the other histone. It's called H1 histone, okay, and forms the nucleosome. Each nucleosome has an octameric, octa that means 8, octa that means 8, okay, octameric core complex made up of 4 types of histone. Two copies each of H2A, H2B, H3 and H4. Around this core is wound DNA approximately 115 base pair in length. Okay, one H1 histone proteins is located outside the DNA on the surface of each nucleosome that complete the structure of the nucleosome. Okay, this nucleosome with about 11 nanometer, okay, is compact and forms the euchromatin and heterochromatin, and then compacted and form the chromosome during the cell division. You can compare the DNA strand. The DNA strand has only 2 nanometer in diameter, and compare the DNA strand to the chromosome. The chromosome has a thickness about 1400 nanometer. Okay, several order of DNA packing occur in chromatin and during chromatin condensation. Some protein like the condensin helps the compact and condenses of the chromatin to form the chromosome. And the other protein is called cohesin. Cohesin protein can attach to chromatid of the chromosome. And usually each chromosome has the small arm and the large arm. Okay. In this picture you can see again the DNA nucleosome, and attention please, you can compare the euchromatin and heterochromatin and the difference of the euchromatin and heterochromatin. In active cell, in nucleus of the active cell, what is the meaning of active cell? The cells can produce the proteins, okay? and has the more activity for production. The chromatin within the nucleus of this cell is euchromatin, and the nucleus is light under the microscope, okay? An inactive cell usually contains the heterochromatin, okay? This picture shows the sex chromatin within the specific cell like the neutrophil in female, okay, in the woman cells. Usually is attached to the lobe of the nucleus of the neutrophil like a drumstick, okay. It's about the inactive X chromosome. We can see this, okay. You know that the X and Y sex chromosomes contains genes determining whether an individual will develop as a female or male, okay? And you know that our cells, or somatic cells, contains 46 chromosome, okay, or 23 pair of chromosome. 22 pair of chromosome are somatic chromosomes, and one pair is the genital chromosomes, okay, like X and Y, okay. And you know that the male the male or man cells contains the XY chromosome, and the woman contains the XX genetic chromosome, okay. And you can see here the karyotype. Karyotype is a map of the chromosome that's arranged under the microscope. You can see the map of the chromosome, okay. And a karyotype provides light microscope information regarding the number and morphology of chromosome in an organism, okay. We can distinguish the dilation of the arm and for example monosomy, trisomy, and the other failure of the chromosome under the microscope. This karyotype is about the men because the sex chromosome are X and Y chromosome. You can see this, okay. And the 22 of the chromosome are homolog chromosome, okay. You can see these chromosomes are homolog, are similar, okay. Each two chromosomes are similar and called homolog chromosome. The other component of the nucleus is nucleolus. The nucleolus is generally spherical and highly dense and highly basophilic subdomain of the nucleus in the cell, okay. And it's actively making protein. It's actively involved to production of ribosomal RNA. Okay. Each subunit of the ribosome, if you remember, it contains the 4 type of ribosomal RNA, and you know that the ribosomes involved to synthesize of the proteins. So the nucleolus, within the nucleus, this structure is involved the protein synthesizes. Under the electron microscope, we can see very large, okay, magnification of the nucleolus, and we can distinguish some area within the nucleolus. For example, pars fibrosa and pars granulosa is about accumulation of the RNA or accumulation of the DNA that's replicated to the to RNA. Okay, so this structure is nucleus. After we learn of the nucleus and its component, we must review about the cell division. Okay, there are two types of cell division: mitosis and meiosis. Mitosis and meiosis. During the mitosis, a parent cell divided into daughter cell. The somatic cell within the body uses to mitosis cell division. Okay, and the other type of the cell division is meiosis, and by the meiosis from one cell, it's called gamete cell. Okay, usually forms 4 gamete cell. Okay, from 1 to N chromosomal cell, the daughter cell becomes N chromosomal during the meiosis. But during the mitosis cell, from 1 to N chromosome, that means the 23 pair of chromosome. Okay, during mitosis cell division, from 1 to N chromosome, 2 daughter cell like the mother cell or parent cell is formed. Okay, so during mitosis, a parent cell divides, and each of 2 daughter cells receives a chromosomal set identical to that of a parent cell. Okay, the event of mitosis can be subdivided into 4 phases. Okay, these 4 phases are prophases, metaphases, anaphase, and telophase. Okay, the centriole separated to each other and moves to opposite pole of the cell, and the nuclear envelope becomes disappear, and the chromatin becomes denser and forms the chromosomes. Okay, during metaphase, the spindle mitotic that formed by microtubule is completed, and the chromosomes becomes dense and attached to the mitotic spindle. Okay, during the metaphase, the chromosome arranged in equatorial plane of the cell. Okay, during the anaphase, each chromatid separates to each other and moves to opposite pole of the plasm. The cytoplasm separate to each other and forms 2 daughter cells. During the telophase, the nuclear envelope appears again, and the nucleolus appears again. Okay, and the 2 daughter cell forms from the one mother cell, for example. Okay, this picture shows the different phases of the mitosis cell division. Okay, you can see each phases, and this picture again shows this process. You can see here each of the chromatid separate to each other and moves to the different pole, and during the telophase, 2 daughter cell is formed. And this picture shows these phases